The definitions themselves give you a lot of hints to putting together these different synthetic questions, such as what are the evolutionary steps for the evolution of the seed? So for example, a seed is technically an embryo with a seed coat and stored nutrition. That's what you may have memorized back in grade school biology or your introductory biology class. But in this class, I want you to memorize that a seed is a mature ovule. Okay, it's an ovule. So for the evolution of the seed, we need the evolution of the ovule. And the ovule is a wonderful structure, okay, that involved then several steps in terms of the evolution of this structure. Now, an ovule typically contains at least one viable sporophyte embryo. Oh my goodness, so the embryo then, or the seed, is an ovule that's containing the sporophyte, right? Not the gametophyte, but the sporophyte. Now, an ovule, this is another important term. Now, the long definition is that it consists of a single megasporangium. Oh my gosh, another botany term, megasporangium. Now, the wall of this thing is called the nacellus. Don't worry about that so much now. We'll come back to that. But that wall then is invested with one or two structures that ensheath it, that surround it. And those are called integuments. So an ovule is a single megasporangium surrounded then by one or two integuments. And the definition that everyone should be able to memorize and regurgitate instantly is an ovule is an integumented megasporangium. Now notice this definition then contains these words that are wrapped up in all of these different evolutionary steps. Integuments, the evolution of a megasporangium, right? Heterospory, reduction then in the total number of spores within the sporangium to just one, okay? One big megaspore within the sporangium is called a megasporangium. Okay, a lot of different botanical terms here, so don't be confused. Go back over your notes, memorize these definitions. Okay. So with that being said, let's now shift gears to our lecture then for today. If I can quickly pull it up here. Excellent. Okay, so for today, given the time, we'll see how far then we can actually get. All right, we're going to try to wrap up a lot of our paleobotany, but in particular, we're going to be focused on the evolution ultimately not only of the seed plants, but a core group of seed plants known as the gymnosperms. The other important group within then the seed plants is a group of plants known as the angiosperms. So two major clades within the spermatophytes, the gymnosperms and the angiosperms. And in order to understand the continued radiation of life, plant life on land, involves understanding what the unique selective pressures then are for life on land, right? It's this increasing colonization of drier and colder environments. All right, so the seed was a key component, but the continued evolution for life on land in terms of the embryophytes involved many other important selective steps and many other important traits, okay? All right, so the most diverse lineage within the vascular plants is the spermatophytes. And so remember, the key synapomorphy to this one big clade, the spermatophytes, is the evolution of the seed, right? But remember, the seed then consists then of an ovule. So an ovule then is a key trait associated with the evolution then of the seed plants. Now, if we look across then the spermatophytes, what we actually have are several other very important traits that are also going to be important synapomorphies. I'm just going to mention them here because it's going to have foreshadowing, right? A little bit of what's to come. Something known as the vessel, another pipe, okay, the vessel. Also, the important synapomorphy of the angiosperm, something known as the carpel, okay? So we haven't quite talked about the carpel yet. Well, actually, we haven't talked about the carpel yet, but we will. It's coming up very quickly. So a little bit of foreshadowing. But then if you notice this whole group of plants here on the side, this entire clade is known as gymnosperms, cycads, ginkgos, coniferophyta, cnidophytes, conifers. A key synapomorphy then of the conifers is that they actually have their reproductive structures in this protective type form. And so there's the structure that protects then the ovule within almost like a cone-like structure. And they kind of differ a little bit, okay? But these cone-like structures where the ovule is then protected, okay? So I would like you to also memorize this phylogeny, okay? And we're going to be coming back to this a few different times. All right, so a seed. Remember, a seed is this mature ovule, right? And it's going to consist of usually one viable sporophyte embryo. It's an ovule that has inside of it this developing sporophyte embryo, okay? Now compare then the life cycle of a plant with an ovule to the life cycle of a plant without an ovule. So last time we kind of ended up with our major kind of quick highway tour of major gymnosperm taxa, the gymnosperms, really cool group of plants. Cycads, they look like palm trees, but they're not. They're not palms at all. Palms are angiosperms. Cycads are gymnosperms. And you can tell, maybe not so much from the foliage or the leaf-like structures here, the leaves, actually these are true leaves. Instead, what you can do is when they're in reproductive stage, you actually see these cones, okay? And it turns out that cycads are true gymnosperms because they only have one integument. Cycads, often confused with palms, they're not, okay? Instead, their seed anatomy is very similar then to the rest then of the seed plants, okay? Cycads are cool. If you kind of slice open this cone-like structure, you'll actually see a structure known as the sporophyll. This is the structure then where all of these sporangia and the ovules are located on the inside. Okay. Ginkgos, another gymnosperm. Ginkgo trees are cool. There's a bunch on campus, and if they enable us to actually have a field trip in the class, it's fun to go out on campus. Maybe we can do this, okay, socially distanced, of course. We can do some botany in the field, which is pretty cool. I can show you some ginkgo trees. It's a gymnosperm. Don't think that this is a broadleaf angiosperm, it's a gymnosperm, okay? Ginkgos are pretty cool because they're actually a living fossil, right? So they were originally thought to be extinct. And we have fossils of them back to about 275 million years ago. Oh my gosh, this was a species, the exact same species that was alive with dinosaurs on the planet, okay? Way back to the Jurassic. Very cool, and they're still around. We thought they were extinct, but they were discovered, a very small population actually in Korea and China. And all of the trees of ginkgo trees are descended from that one population that they actually rediscovered. They thought we were extinct. All right, another group of gymnosperms, really cool group, gametophytes, Gnetophyta. So the G here is silent, G-N-E-T-O, Gneto, all right, Gnetophytes. And so there's actually 3 extant genera. They're kind of cool. They have vessels in their wood. We're going to come back to that. And several Gnetophytes, especially if you study tropical forests, in particular cloud forests, they tend to be these lianas. Now, a liana is a term for a woody vine. Okay? And a woody vine means their stem actually gets really woody and they get really big. And so you find these things, we can be doing either kind of surveys or doing field biology way up in the cloud forests in some great locations. And we discover then these things, we're like, oh my gosh, it looks like a great angiosperm broadleaf plant. But actually, if you look for the reproductive structures, there are these cone-like structures. And the ovule only has one integument. It's a gymnosperm. Gnetophytes. The coolest gnetophyte, in fact, my vote for the coolest plant on all the planet is Welwitschia, another gnetophyte. Here it is growing in its natural habitat in the deserts in Namibia in Africa. Look at this thing. It's like these leaves are like vomited on the ground. It's all disheveled and oh my gosh, it looks like this alien plant. These things are so cool. You go out and see these things. It's like nothing else living, all around, but there's this Welwitschia. And what's cool is that if you wait around for the reproductive structures to show up, there are these cone-like structures. They kind of look like a little pine tree, little pine cone on the inside there. And so we call these structures strobilus. So there's the female strobilus and the male strobilus. It's a gymnosperm, okay, that's one integument. All right, cool plant. Another cool one you actually find here in Arizona, here's another neophyte, ephedra. If you go out to Saguaro National Park or even the desert foothills here of Tucson, you'll see ephedra growing. And sometimes you'll see the little reproductive structures, little cone-like structures, little cones, ovule, one integument, right? You know it's a gymnosperm. Okay, but what everyone loves in terms of gymnosperms, the conifers, right? Pine trees, fir trees, spruce trees, cedar trees, all with these cone-like structures. Ooh, look at this. That doesn't look like a cone. That looks like a juniper berry. Actually, it kind of is a cone-like structure, and it's not a true berry. In fact, that is a convergent berry-like structure. We'll come back to talking about interesting juniper berries, but it's still a gymnosperm, one integument. All right, so why seeds? Why did we have selection, all right, across the board for seeds? And then why then were seeds necessary to basically be then the critical trait that enabled colonization for a good chunk of the surface of the land on Earth? All right, so here's a great question for the midterm exams. Can you articulate and kind of clearly set up the logic for the main selective drivers for the evolution of the seed? Okay, so I had you remember at the start of class the essential steps for evolution by natural selection. Okay, and so when I ask you a question like this, I would emphasize to you, go back to your memorization of those essential steps for evolution by natural selection. And also now take into what you know about successful survivorship and reproduction on land. Okay, can you articulate the main selective drivers for the evolution of the seed? All right, so I'll give you an answer here. So there has been extremely strong selection for increased colonization of environments where no other plants were living, right? If you had the traits where you could survive in a place where no other plants are living, there would be no competition for resources from anybody else. You would have the entire place to yourself, right? And you would have all of this open resources then for your potential offspring to utilize. Okay. So there was strong selection to increasingly colonize more dry areas of land because there are no plants there, right? If you could survive and reproduce there, gosh, all right, that's what selection does. So they're increasing selection to colonize then these locations. So think about this again in terms of fitness, survivorship, and reproduction. So increased survivorship and reproduction then is favored then in these drier environments, right? And again, because it's drier, there's strong selection to eliminate the requirement of water to fertilize eggs. Because in dry environments, water may not be present, if at all. Right? So how then does selection then deal with that? Okay, what traits then are favored? But increasingly, because of increased aridity, okay, and also in colder temperatures, embryos had a hard time surviving. How can we increase the survivorship then of embryos? Protect, again, from desiccation, lower amount of resource availability, and so on. And so one solution to that is the increased provision of resources to the embryo, right? So this then increased survivorship of the embryo. Get more maternal investment, more supply of resources than from mom, you can increase then the survivorship of your developing embryo. So again, more resources to withstand increasingly harsher climates, increasingly harsher abiotic environments. So again, seeds then are the nourishment for then this developing embryo. And so their eggs, okay, then are also increased then survivorship. So the sporophyte generation, again, retains then the egg and embryo just like a female animal does. This is convergent evolution associated with maternal investment. Okay. You see this both in plants and animals. Okay, so I asked you then on the Jamboard, what's the reproductive syndrome that all seed plants involve? So this reproductive syndrome then is a series of steps, okay, selective then steps for specific traits and specific behaviors then in plants to increase in order to increasingly colonize dry environments. retention of megaspores within the megasporangia, the reduction then the number of megaspores per megasporangium to just basically ultimately one functional megaspore. A lot of resource then that's allocated to ultimately that developing embryo. We're then going to see additional selection to enclose that megasporangium, that structure, that reproductive structure, by additional structures known as integuments. And once we get this kind of wrapping, this ensheathing then of the megasporangium by these additional fleshy layers, those layers are known as an integument. Once we have a megasporangium surrounded by an integument, that's an ovule. That's the definition of an ovule. But then we also have selection for a modification of the megasporangium itself to actually receive then microspores, okay, more the male-like function, and the sperm-bearing microgametophytes, okay? So all of these features evolved relatively early. Okay. So the first evolutionary steps or transitions then to the ovule, we can see this in the fossil record, all these different fossil organisms then that had these kind of fleshy additional structures surrounding then on the inside the megasporangium. So on the inside here, we actually have the megasporangium, and on the outside, we have these fleshy kind of additional structures on the outside. So on the inside of all of these, okay, we actually have the megasporangium. And then once we get this wrapping, this enveloping then of these kind of outer structures of then the megasporangium, then we have an ovule. So the integuments evolved then from these fleshy lobes that surrounded then the sporangium, okay? And so the gradual fusion over time, okay, provided this additional, the maternal protection layer, okay? And because of which, we now have a structure known as the ovule. All right, so the earliest fossil seed plants, okay, these early gymnosperms are actually found in the Devonian, the late Devonian. Again, remember all the action happens in the Devonian. And so these gymnosperms are then kind of consisting of these fleshy lobes, okay, these structures known as the salpinx, okay? This is a fallopian tube. And this is a tubular extension, okay? And so these fleshy lobes that have this tubular extension then on top were likely specific structures selected for to help kind of trap the microspores, okay, in terms of kind of not only trapping them, but then kind of increasing the probability of fertilization. Okay. And so this tubular extension probably helped in kind of trapping these microspores or these pollen grains, right? And kind of facilitated more fertilization by wind. And so this salpinx then was likely the structure that ultimately gave rise to integuments or the ovule. Okay. So all of these plants that we know of in the fossil record, they're all extinct. All right, so here's one example, kind of reconstructed then from rocks, from fossils, and showing effectively the steps to the development, evolution of the integument. Okay. So we have these fleshy structures known then as this cupule. And so we have these hair-like structures then extending out, probably helped in trapping and attracting to some degree, thanks to hydrodynamics, fluid dynamics, all these microspores out of the air and basically dropping them right on top then of the megasporangium, where we have one functional megaspore. So all of these microspores landing in close proximity then to the megaspore increase then your fertilization possibilities. Okay. So these structures, these hair-like structures then extending then above, right? So we see that within Elkinsia here, we have the salpinx structure with these hair-like structures up above. A lot of the early gymnosperms had then this structure. And this is likely a specific adaptation for the attraction and trapping then of microspores. And so we kind of give this analogy of a snow fence. And so if you've been to northern Arizona or places where it snows a lot, they put out these snow fences around either houses or highways to trap then the blowing snow. And on one side then of the fence, all of this snow then that's blowing through the air gets deposited on the other side, and it kind of builds up, okay? And the reason why is that the wind is kind of cruising along and the snow is blowing and it hits then these open fences, right? and it slows the wind down enough and creates a little turbulence so that the snow is deposited actually on the other side. So you can actually attract snow to a given location by kind of forcing it downward, and you build up then these big kind of snowbanks on the leeward side of these things. Same physical principles in these early gymnosperms. Basically, wind kind of blowing through, you have these microspores of early pollen. It's kind of entangled to some degree and it creates some vortices and some uncertainties on the wind, and that kind of drags then a lot of these particles out then of the wind stream and forces it downward, kind of increasing the likelihood then of a fertilization event. Okay, so we see within the fossil record these very clear kind of steps in terms of the development then of the ovule, where we have the sporangium, then we have fossils then that have sporangium with both small spores and big spores, microspores, megaspores, and then ultimately then we see sporangia with just functionally one megaspore, okay, on the inside. And then we see fossils that have not only a megasporangium, but we start to see additional structures, these kind of cupule type salpinning structures that begin then to encapsulate the sporangium itself, okay, containing the one megaspore. And once we have that, that's the ovule, okay, where we have inside, we have one functioning megasporangium with a megaspore, okay, so a megasporangium, and then on the outside we have this integument layer, and together that forms then the ovule. All right. So within the gymnosperms, we also see these other reproductive structures, these cone-like structures, and we call these cone-like structures a strobolus. Okay, that's a weird term, a strobolus. And the plural of a strobolus is a strobili, right? Strobili, strobolus. And it's an organ, it's a reproductive organ, okay? And it's usually kind of surrounded, kind of, well, characterized by being on top of this central axis. And so here's equisetum, okay, a really cool gymnosperm. And you have these spirally arranged structures here, okay? And so a strobolus then is a structure then that contains all these different reproductive structures, okay? Ovules, bunch of different ovules, right? And so once we get within the conifers, pine trees and fir trees, strobili are called cones, okay? So a pine cone is a strobolus, okay? And a strobolus in a conifer is known as a cone, all right? All right, cool. So within the lycophytes, sphenophytes, cycads, gymnosperms, all right, we have these structures known as the strobolus. All right. Gymnosperm. All right, so like a lot of botanical terms, it's really great to kind of break down these terms. Okay, so gymno, literally meaning naked, sperm meaning seed. So gymnosperm literally means naked seed. Okay, and so do you know what gymnasium means? Gymnasium, thanks to the Greeks, is known as the house of nakedness. Go figure. But hopefully now you'll memorize what gymnosperm actually means. Gymnosperm means naked seed, and it means naked because the ovules are exposed to the air, okay? They're exposed to the air, all right? They're not fully covered yet, okay? So gymnosperm, naked seed. And the ovules themselves are actually located in the open on top of a flat, something known as a sporophyll, okay? And a sporophyll in a cone is actually this little bract. Each of those little bracts coming out, those are known as sporophylls. And the ovule sits right on top of that sporophyll. All right, now I'm foreshadowing here because this is a more botanical definition. So within a gymnosperm, the ovules are not enclosed, they're not hidden, they're not covered up by something known as a carpel, all right? But on a gymnosperm, they're open, they're exposed to the air to receive then pollen, right? So a gymnosperm, a naked seed. Okay, so I would like to now start adding to your life cycle memorization, right? Your life cycle, the evolution of life cycles and with the land plants. And so we can jump ahead to a gymnosperm cone, in particular a conifer cone. And so cone evolution is associated with increased protection of the ovule, Okay, so a cone is nice and woody, right? It's kind of hard to kind of get inside, but still, if you open up one of those scales or one of those bracts, okay, one of these bracts here, here's a pine cone, then that's sliced right down the top. And so you actually find the ovules are actually sitting right there. So there's an arrow, there's an ovule right there, there's an ovule right there, okay? So if you start breaking open pine cones, you can actually find the ovules sitting right there, kind of right at the base, right? And so it has then this big woody structure, okay, for increased protection. And so you can find male cones, okay? On the male cones, you actually find these microsporangium. And on the female cones, you'll actually find the megasporangium, but now we call it the ovule. So remember, the ovule consists of a megasporangium. Now on the male cones, they then released, okay, these microspores, but now the microspores are known as pollen. Okay? Microspores are then released, they blow then by the wind, and ultimately, hopefully, the wind will then blow it into the cone, the female cone, in close proximity then to the megaspore or the ovule. Okay. So the development of an ovule. Within a pine cone, we can go through the different steps, okay? So here we have an ovule, okay? And on the outside, we have this structure, this ensheathing structure known as the integument. And on the inside, we actually have several megaspores. And so within the gymnosperm, we have actually kind of these 4 megaspores then that are kind of formed. But then we also have, I'm kind of click through here, an abortion event happens where these 4 megaspores then that are formed from meiosis leave behind only 1 functioning megaspore. Okay, that megaspore then will develop. Okay, it's going to then divide, okay, or be infused then with resources. I'm sorry, it'll be kind of grow in size, and it will then develop an archegonium. Okay, so we actually see the development then of an archegonium. Okay, so remember the archegonium is that on the sporophyte or the gametophyte. So this wall-like structure then that's surrounding kind of all of the developing megaspores is something called the nacellus, okay? And that's going to be very important in terms of where resources then come from for the developing embryo in the seed. So this is very different from earlier embryophytes, okay? So now we have the gametophytic stage totally enveloped and kind of formed within then the sporophyte itself. So remember, this is happening all within the sporophyte. So now the gametophytic side of the life cycle is within the sporophytic side of the life cycle. Kind of bizarre and crazy. All right, so the megaspore develops into the megagametophyte inside the sporophyte. Okay, so we haven't lost the megagametophyte yet, but instead it's within the sporophyte. All right, so let's kind of continue on then our life cycle in a gymnosperm. Here then we're going to have the microspore, now known as pollen, kind of trapped then and kind of landing on top then of the megasporangium or the ovule. So we have ultimately a fertilization event. Okay, so a pollen grain is then going to land, right? That pollen grain is known as a microgametophyte or a microsporophyte. And these microspores then have to pass then through the maternal tissue. And this is very important because this is a point then on the maternal side where we can have maternal control of fertilization events. Okay, so whether or not the megaspore is then available and receptive, okay, to a microspore or not. So we have to then have the microspore actually pass through the maternal tissue, right? And so this is where pollen tubes then come in. Now, as soon as this microspore makes it to basically the entrance then of the archegonia itself, we have what's known as a pollination droplet forming. Okay, This is a little droplet of water then that's formed right at the entrance then of the archegonium. And that's important because sperm is delivered then by the microspore, and sperm still has to swim this last little step, okay? So we have this last little requirement of water, but occurring then within the sporophyte upon then entrance then to the gametophyte. Sperm then swims, okay, from then the microspore into the archegonium. We then have a fertilization event. And the zygote then develops via mitosis within the gametophytic tissues, okay? So if you notice then the ploidy levels then here, we're gonna be going from an outside ploidy level of diploid. Once we hit then the ovule, that's mainly gonna be haploid, but once the zygote then is developing, that is then diploid. So that's, I know that's a little confusing. So it's important to kind of like go step by step then through this. Okay. Now, the integument then that's surrounding then the megaspore itself, upon fertilization, the integument then develops then into the seed coat, okay, the actual coat of the seed. And so then the seed is containing this sporophyte embryo. All right, so that's a lot, all right? And so what I would like you to do is try to form either a study group, or if you're really good studying, with yourself, go through these steps, okay? And so you'll likely see some component of this on the midterm exam, because remember, I'm asking you to memorize, or at least be able to functionally label and understand the evolution of the life cycles then, from green algae all the way up now to the seed plants, okay? All right, so seed development. After fertilization of the ovule, the zygote is gonna continue to develop then within the integument itself. And the sporophyte embryo is going to grow and mature within then the megagametophyte. But ultimately, the integument then forms the outer seed coat. Okay, really cool. All right, so go check out some seeds. And what you're looking at is this mature integument, right? That was once then kind of the outside, well, the key component then of an ovule. All right, so evolution of seed plants is the continued reduction of the gametophyte. To basically the gametophyte is being reduced and reduced and reduced to the size. And again, compare this with early land plant life cycles. The megagametophyte is now entirely retained within the megasporangium. There's a lot of megas there. Megagametophyte is retained within the megasporangium. Right? I love it that botanists love mega. It's like all over the place. All right. Now, the integument of gymnosperms, it turns out, can be fleshy, or they can be hard, okay, or dry. So you likely know most seeds, okay, have this kind of outer, kind of dry and hard kind of seed surface, okay. So that's a hard, kind of mature integument. Now the integument can also be fleshy, right? So if you're familiar with yew berries or juniper berries, okay, if you know where gin comes from, right? They make gin out of juniper berries. Okay, berries is in quote. It's actually a bad botanical term because it's not even a berry, not even close. But that fleshy component of a yew berry or a juniper berry is a fleshy integument. That's the integument. So the integument can be kind of forming this hard kind of outer then layer outside then kind of forming the outer surface then of the ovule, or then it can be this fleshy outer layer. And this is the first time then in the fossil record where we start to see evidence of coevolution with animals for dispersal then of seeds. Fleshy integuments. These yew berries, juniper berries, certain birds and other animals actually love to eat these things. And in the process of doing so, pass them through their digestive tract and out the backside of the bird, fresh with a lot of fertilizer, ready to germinate. Okay, we'll have more in coevolution as we come up. Okay, so pollen grains. This is the first time we've mentioned pollen. So pollen is really interesting because it is a structure then that kind of contains and houses these microgametophytes, and it's almost like a coarse powder-like structure. And it consists then of these pollen grains, these microgametophytes. And again, pollen is what's going to be carrying the male gametophytes of seed plants. And so each pollen grain consists actually of two cells, two generative cells, two male gametes, okay? And also a vegetative cell, all right? So the pollen grain itself actually contains several living cells, either one or two generative cells and one vegetative cell. And so in total, there are going to be three then cells within then a pollen grain. And so the pollen grain itself, again, is this, it's housing then these three living cells. And so this group of three living cells is then surrounded by a cellulose cell wall and a tough outer layer made of sporopollenin. All right, so this isn't the first time you've heard sporopollenin. We talked about sporopollenin as a key component then for the evolution then of cuticles for the land plants to withstand desiccation. So here we have sporopollenin being deployed then by plants, selection then to encase then these microspores to make sure that they don't desiccate these living cells. So pollen is this vehicle, think of it as this little spaceship containing these three living cells. So pollen is living, okay? And as it blows then around, right, it's really prone to being desiccated. Because it's small, it has a high surface area to volume ratio, meaning that it can dry out really quickly. There's a lot of surface area. And so the sporopollenin retains then the moisture of these three living cells on the inside. And so pollen can actually live for a very long time. And so the evolution of pollen in the pollen tube and the seeds together, this marks this critical turning point in plant evolution. Because we have all of these different steps, we have all of these different components then together, definitely by the Carboniferous, okay, which is now coming out of the Devonian about 358 million years ago. Now, some gymnosperms evolved microgametophytes then that formed additional structures known as pollen tubes that actually grew then out of then the pollen to then deliver then the male microspores directly then to the female archegonium. And so these pollen tubes actually grew then through then the nucellus, through then the maternal tissue. And the evolution then of the pollen tube ultimately eliminated the requirement of water for sperm then to swim, effectively making plants independent of water. Wow, it took that long, okay, but they finally did it. They finally broke their connection then to water and sperm having to swim. So then with the rise of the angiosperms, they actually kind of reconfigured life on the planet because the gymnosperms had all of these different traits that enabled them to break free from water and to be a superior competitor than to all of the other plants then that were present on the planet at the time. And because of which we start to see at that time this big change in the taxonomic composition of life on land. So beforehand, we had these earlier embryophytes, and in particular, quite a few kind of fern kind of species and other bryophyte species, and also lycopods. But with the rise of the gymnosperms, we actually see that the gymnosperms were a superior competitor. And they started to replace the rest, a lot of, well, not the rest, but a good chunk then of the vegetation of the planet. And they became the dominant terrestrial vegetation because they were a superior competitor. They had this new technology, right? The seed. It enabled them to increase their survivorship and increase their reproduction. And because of which, they also were able to colonize increasingly drier parts of the planet. All right. So this immature microgametophyte of a seed plant then is the pollen grain. And among the seed plants, it's this microgametophyte then that develops then the pollen. And pollen then, upon landing on the female tissue, then develops then this extension known as a pollen tube to actually deliver very effectively the microgametophyte right to the archegonium. And so the pollen tube then grows through the nucellus, grows then through this female tissue to the archegonium and deliver sperm cells directly to the megagametophyte. So here's the life cycle of a generic gymnosperm. Okay. So we have the mature sporophyte, okay, your pine tree or your fir tree. We have then these reproductive structures known as a strobilus, okay, or a cone in the case of a pine tree or a fir tree. We have our ovulate cone. We have both male cones and female cones. On the female cone, we have the ovule sitting then on the cone bract or the sporophyll, receptive then to pollen. Okay. On the male cone, we have microspores then released. Okay. Again, we're having meiosis then producing then the egg, meiosis then producing then the sperm, right? We're then releasing then these haploid gametes, be it then an egg, okay, or pollen grains traveling through the wind. The wind is then blown in, and hopefully, the pollen is then blown by wind, hopefully landing up then in close proximity to the ovule. And if that is the case, we'll likely have a fertilization event where the pollen tube then delivers then these regenerative cells, the reproductive cells, these three cells, they together then form pollen tubes, drill then down to the archegonia where we then deliver sperm directly, have a fertilization event. And in the fertilization event, we then have the developing sporophyte within the embryo, okay, and that develops then within the megagametophyte, all on the inside, okay, to form then the seed. Seeds are then released, dispersed, and now we have our seedling, our developing sporophyte. Okay. Generic life cycle of a generic gymnosperm. Cool. So what I would like to end with is a very important but mysterious term known as the leaf, and what the leaf then can tell us about filling in these next important components of the evolution of life on land, on plants. So we're going to go back to paleobotany and fill in a few gaps here. What is a leaf? Earlier vascular plants. You know what they look like? Really dinky. No leaves. Green photosynthetic stems, stomates on the stems then themselves. Reproductive structures, sporangia on the tips. Early vascular plants. But around the time of the late Devonian, we have this split in land plant evolution. Land plants are biphyletic. Remember, we have a group of plants known as the Zostrophyllophytes, my favorite word, the Zostrophyllophytes, and the Trimerophytes. The descendants from the Zostrophyllophytes then gave rise to an entirely different branch than of land plants, whereas the Trimerophytes gave rise to a whole other different group of land plants, including ferns, gymnosperms, and angiosperms, including the spermatophytes that we just talked about. So now let's look what happened on the other side of the phylogenetic tree. What about this other phyletic branch? What about the descendants of the Zostrophyllophytes? So lycophytes turned out dominated. This was the group of plants, the Zostrophyllophytes, dominated during the Carboniferous. So if you look in the Carboniferous, the descendants of the Zostrophyllophytes, the lycopods in particular, dominated. Whereas the descendants of the Trimerophytes were not that present. They were there, but they weren't really too dominant. But if you notice what happened over time, they took off and ultimately dominated and outcompeted this other set of descendants from the Zostrophyllophytes. It's a sad tale. It's a tale of two houses. One grew in dominance over the other, but then the others acquired certain traits that allowed them to dominate and take over basically most of the planet. Okay, so this is the sad tale of the Zostrophyllophytes and the lycophytes. The lycophytes dominated the Devonian and the Carboniferous. Really cool trees. They comprised actually 2/3 of all plants within the Carboniferous. They were everywhere. They're really common in swampy, wet environments. But sadly, there's only 5 genera living today. You can still find these descendants of this dominant group of plants known as lycophytes back in the Carboniferous. But sadly, they're pretty small. Club mosses, Lycopodium, you can find these guys in Saguaro National Park, east and west, within the Catalina Mountains. After it starts raining in the wintertime, if you go to low elevations, all this kind of green around the rocks, it looks like a moss, but it's actually a lycophyte. You can find them here in Arizona. So this is a tale of what a leaf is. Because extinct lycophytes and extant lycophytes didn't have true leaves. Their leaves were called microphyls. They're not true leaves. This is a different independent evolution of a leaf-like structure. A really cool dominant plant, something known as lepidodendron. Actually, my hard drive is called lepidodendron. Right? It's a cool tree. They had sporangia within cones. So the evolution, independent evolution of cone-like structures, strobili. So these reproductive structures which contain a high concentration of sporangia. Here's another one, Sigillaria. Really bizarre plants. Monopodial growth, basically main central stem and this explosion of disheveled branches and leaves, these microphyls all on the top. So their leaves are called microphyls. They're not true leaves. Monopodial growth. They grew upward from a single meristem and then kind of exploded like a party favor right on top. Really awesome fossils, all right? Lepidodendron, okay? In your own time, you can kind of check out some of these pictures and check out some of these links. We have really great fossils of these guys. In fact, you can basically go into a fossil lepidodendron forest. We have entire trunks and stems and everything. You can kind of walk through to look at like what these forests used to look like. Really cool stuff. Look at this. You can still find the stumps and the roots. Oh, roots. Now we can start talking about roots a little bit. We'll come back to roots. Really cool lepidodendron fossils. Here are the leaves, but they're not true leaves. They're called microphyls. Another word is called a nation, but they're microphyls. They're not a true leaf. And we've been able to reconstruct these things, okay? And so this is what these forests used to look like. Again, wet, swampy areas, right? Look at this, beautiful forests of lepidodendron and sigillaria. Really awesome. These things are bizarre. It's like a foreign land, right? And you can find most of their root structures already intact and fossilized because they tend to grow in these anoxic conditions, right? Where things were preserved. What is that? What is that? It's an animal, gosh darn it. They start to come into play here around the Carboniferous. That's ignoring animals for a little bit. But for the most part, the plants had the world to themselves. Lepidodendron, sigillaria in the Carboniferous. So what's great is that we can now start to take what we know about these fossils and we can reconstruct these landscapes, what they look like. And so here are some of these gamers, and they actually paired up with a bunch of botanists, and it's fun. You can kind of cruise around. There's several sites that if you want to kind of go around the Carboniferous and look botanically what it was truly like, right? You can see, oh my gosh, you can see some that looks like some equisetum there. We have some sigillaria and lepidodendron. Oh my gosh, you still have some green algae growing around. Yes, they're still around. A Spermatophyte. So we have ferns, okay, some fern-like structures all around. We have awesome fossils. In fact, a lot of these fossil forests were pretty much preserved intact because in these environments, they're often prone to flooding and these big inundation events that basically buried and kind of fossilized everything today. So you can check this out kind of on your own time, but we have a really good idea of what these environments used to look like. But you know what now, what we call all of this vegetation? We call all of this vegetation coal and oil, right? All of that ancient photosynthesis that used to build all of these structures, all of this wood, right? When these plants died, they fell into these swampy environments that was very anoxic. So there wasn't a lot of decomposition or respiration. And over millions of years, all of these trees in these swampy environments fell over and was preserved. All that carbon was locked away. All that ancient photosynthesis then that's occurring right there, locked away and buried under successive layers of flooding and sediments, and ultimately increasing sediment layers and rock. Right? Millions of years of this, of all of that photosynthesis in leaves and trunks buried in these swampy environments. And today, when we dig those up, okay, all that ancient carbon is in the form of coal, oil, and natural gas. Okay, that's all ancient photosynthesis. All right. So this brings up the tricky issue of what a leaf is. And it turns out that leaves can evolve multiple times. Right? They have evolved multiple times, leaf-like structures. So one leaf structure that evolved is something known as the megaphyll line of plant evolution. This is a true leaf. And so if you imagine our initial Cooksonia, this dichotomous branching structure with these green photosynthetic stems, there was selection for increased photosynthetic rates. And one way to do that was to increase your surface area by increasingly adding on more kind of web surface area, more photosynthetic tissue than off the stems. And once those kind of overlapped and formed, right, we have then the evolution of what we know as a true leaf or a megaphyll. So what you know of a leaf, if you go out and pull off a leaf off of a random plant, most likely it's a megaphyll. And a megaphyll is developmentally very different from a microphyll. So a megaphyll then includes this underlying branching structure and this webbed lamina or this photosynthetic lamina itself. They develop together. Okay? So the stem, the central stem and the lamina together develop together with each other. So the leaf venation is actually this internal kind of structure. You're looking at the kind of ancient paleo equivalent to Cooksonia and its dichotomous branching. So leaf venation is reflecting that early land plant dichotomous branching in the megaphyll line of evolution. So this is a true leaf, a megaphyll. Now the megaphyll line of evolution ultimately gave rise then to the seed plants. And there's a lot of different groups. Don't worry about these different terms. Okay, if I haven't emphasized them, now I'm not emphasizing any right here. Don't worry about that. But there's a lot of different groups kind of associated with the megaphyll line of evolution. But they're all coming out of the Trimerophytes. So the first plant with true leaves or megaphylls is something known as Archaeopteris. This is the first true forest plant with the true leaves, a megaphyll. And it appeared then within the late Devonian. Remember, I told you all the action happens in the Devonian. And so Archaeopteryx then descended then from a line of ancestors then from the Trimerophytes. Okay. This is the first kind of true leaf megaphyll tree. So Archaeopteryx, this is a pro-Gymnosperm, one of the first dominant trees on Earth. Okay. Remember, this is evolving then around the time after then Lepidodendron and Sigillaria, okay, from the microphyll line of evolution evolved. All right, so we have really good fossils of these guys too. So these early seed plants consisted of pro-Gymnosperms, kind of this typical apical dominance growth form. Okay, seed ferns, which is a horrible name, all right, because they didn't actually truly have seeds yet, but they bear ovules then on these leaves. And so by the Carboniferous, we had 5 major plant groups. Okay, so between 363 and 290 million years ago, within swamp moist forests, we had lycopods, sphenophytes or horsetails, pteridophytes or ferns, pro-Gymnosperms, and these early seed plants. And so these plants then consist of plants either with microphylls, lycopods, or true leaves, megaphylls, such as these early seed plants. Now, many of these, okay, are extinct, but some of these groups still have descendants left behind. That is, some of them are extant. So a Carboniferous forest then consists of these wet, swampy forests and some of the first animals, insects, early reptiles. Today, we call most of this carbon fossil fuel, okay, meaning ancient photosynthesis. All right. So with that, I think I'm going to end it there, and I'm going to leave you with a very nice view looking out on the Carboniferous. Okay. So we'll return on Thursday.